A warm welcome to the Integrated Environmental Health Sciences Seminar Series. This binary series welcomes members and guests of both the P30 Center for Urban Responses to Environmental Stressors, CURES, and the P42 Center for Leadership in Environmental Awareness and Research, CLEAR, both funded by the National Institute of Environmental Health Sciences. I'm Graham Parker, the leader of the Translational Research Support Core of CURES, and it is my great pleasure today to introduce our guest speaker, Hilary Marusak, Associate Professor of Psychiatry and Behavioral Neurosciences here at Wayne State University School of Medicine, and a researcher on the CURES Resilience Research Interest Group. Hilary's research advances understanding of cannabis and the endocannabinoid system and their effects on brain development and mental health. Hilary also uses neuroimaging, behavioral, and physiological approaches to study neural mechanisms underlying stress-related psychiatric disorders. Following a bachelor's in biology and psychology at Kalamazoo College, Hilary pursued a PhD in translational neuroscience here at Wayne State under former CURES researcher Mariah Thomason on the neurocircuitry of pediatric emotion processing before completing a postdoctoral fellowship in the Translational Neuropsychopharmacology Laboratory of Christine Rabenak in the Eugene Applebaum College of Pharmacy and Health Sciences. One day later, she joined the faculty of the Department of Psychiatry and Behavioral Neurosciences here, becoming the director of the Trauma History Investigation of Neurodevelopment in Kids Lab. Hilary has earned a cascade of honors and awards, including the prestigious Rosalind Franklin Society Special Award in Science. A fierce advocate for community engagement, Hilary is the co-founder and faculty advisor for the Science Policy Network, bridges the gap between scientists, lawmakers, and the lay public. Hilary has no less than 8 pages of mentorship entries on her very small font CV. She is currently funded by the National Institute of Mental Health, the National Institutes of Child Health and Human Development, the State of Michigan Department of Licensing and Regulatory Affairs, and the One Mind Mignana Lovelace Rising Star Award in Mental Health Research, as well as holding a current CURES pilot project grant, which seeks to improve health outcomes for children living with caregivers who regularly use cannabis. In today's seminar, Dr. Hilary Marusak will engage us with her presentation, Adolescent Mental Health in Context, Trauma, Air Pollution, and Cannabis Effects on the Developing Brain. Welcome, Hilary.
Thanks so much, Graham. Now I feel like my work is over with. You just told my whole story already for me, so thank you. So I know some of you are eating lunch, but I'm going to put you to work in a moment. And I wanted to start with a statistic. Did you know approximately 1 in 4 adolescents experience a mental disorder within the past year? And a majority of these mental disorders begin during adolescence. What if I told you that the environment they're exposed to, the air that they breathe, the experiences they have, and the substances they use can shape their developing brain in ways that make them more susceptible to mental illness? So again, I'm going to put you to work. If a couple of you want to unmute or just put your answers in the chat, I want to ask you this question and have you start thinking about how these environmental exposures can affect brain development. So go ahead and cast your vote. Again, you can unmute, you can put it in the chat. Which of these do you think has the greatest impact on the developing brain? Trauma, air pollution, substance use, all of the above. I'll give you all a minute. Graham, you are unmuted. Do you want to share your response?
Oh, I was going to type it in the chat and try and be polite, but I think it's D, isn't it?
Okay. We're getting some A, D, A, all of the above. Yep. You guys are mixed. Lynn, yeah, so I'm not going to directly answer that today, but I love the votes. And just to give you a little bit of a lay of the land of what I'm going to talk about today, I want you to leave this talk understanding adolescence, how and why it's a critical period of neurodevelopment and a time of great psychiatric risk. And of course, I don't have time to talk about each individual exposure, but today I'm going to highlight 3 key environmental exposures that my lab has been working on in this area, specifically trauma, air pollution, and substance use, particularly cannabis use. And then I want to end on a high note and give you a little bit of a taste of what we're doing in the biomarker identification as well as the intervention space. So let's start off with brain development, the basics. I am a developmental neuroscientist, as Graham mentioned. Most of my research focuses on little people, on children and adolescents. And I know Chris is in the room, so you can probably relate. We don't do this just because we like to make our lives hard and keep kids still in an MRI scanner for an hour or so. Pro tip, just pay them a lot and they will do that for you. But no, I don't study children and adolescents because I think they're adorable and hilarious, which is also true, but because many of these psychiatric disorders, some of the most common mental disorders, begin very early in life. So what I'm showing you here is the age of onset distribution of anxiety shown in red and some other common disorders. And what you'll see right off the bat is that many of these disorders begin within the first 2 decades of life. And in my lab, we frequently focus on anxiety as it's a sort of a harbinger of later depression, substance use disorders, and other mental disorders. So in addition to adolescence being a significant period of psychiatric risk, it's also a period of substantial brain development. And some of you have seen this graph before, but I want you to have the take-home message that adolescent brains are not just like shrunk down versions of adult brains. And for those with kids who have ever been around children, you know that that's not the case. It's an entirely different landscape. So on the left here, I'm showing you a sort of timeline of gray matter changes across the childhood and adolescent period. And what you'll see is that different brain areas mature or lose gray matter density, which sounds bad, but we think of it as the brain becoming more efficient, shown in more darker colors. The first take-home message is that different brain areas mature at different rates. So it's a changing landscape, so to speak. And on the left, the right side of the brain is the prefrontal cortex, the area right above your eyeballs. On the right side, I'll show you an animation where the prefrontal cortex is on the top, so kind of a top-down view of the brain. But in addition to different brain areas maturing at different rates, the second take-home message is that that prefrontal cortex, again, the top of the brain, is one of the last to mature. So I'll play the animation for you. Let's see if you can notice it. There you go. So again, that prefrontal cortex is really important for emotion regulation, for impulsivity regulation, decision-making, future-oriented thinking. Again, think about the things that adolescents are learning, let's just say. So all this flux during adolescence makes it a particularly sensitive window for environmental insults to have dramatic impacts and for things to go awry in things like psychiatric disorders. And one of the insults that I've studied throughout really my entire career is childhood trauma exposure. So that could be violence, abuse, motor vehicle accidents, natural disasters, losing a loved one unexpectedly, like in the case of COVID in some cases. And unfortunately, these experiences are really common, especially in urban environments like Detroit, Michigan, where in some of our studies, 90% of youth have experienced trauma. And again, I'm sure a lot of folks on the call that work with similar populations have seen similar statistics. And actually, the norm is that most children have experienced more than one trauma. So that is a lot more common than just a single event. What I'm showing you here is Detroit rates of trauma exposure, as well as some common psychiatric symptoms or disorders against the national average. And unfortunately, Detroit is highly, we are winning, not in a good way. We're exceeding that national average. And this is, again, really common, especially in urban environments like Detroit. So one of the things that we've been studying is how trauma shapes the brain, because we know trauma exposure is one of the most significant risk factors for mental disorders, as well as physical conditions like hypertension, diabetes, obesity, and cancer. And we've used neuroimaging to chart how childhood trauma exposure changes brain function in regions and networks that are involved in regulating emotions, including fear, which is really relevant for anxiety and PTSD, as well as core attentional processes and other high-order cognitive functionings. So we're thinking about this as trauma changes the brain in ways that make some people susceptible to later disorders. And although today, luckily, I think this idea isn't new for a lot of folks, that the environment and experiences we have can shape the brain and make people susceptible to psychiatric illness. But even a couple of decades ago, and when I was beginning this work, this was really a new area. There's a lot of interest in looking at genetic risk factors for things like depression, like having a first-degree relative. But the notion that the environment can have such a dramatic impact on the developing brain was relatively new. So along the way, we've also expanded our scope of what we consider to be trauma and looked at different populations. And one population we came to work with were kids that have been diagnosed with cancer. And this was kind of a serendipitous project that came when we met Jeffrey Taub, who is a pediatric oncologist at Children's Hospital of Michigan. He's also a cancer survivor himself. And he saw in his patients a lot of what looked like PTSD symptoms. Kids were startling, they're very jumpy when things happen, when loud noises happen. They re-experienced or had nightmares or flashbacks about being in the hospital and receiving port start procedures. But there wasn't a ton of understanding on the psychological impacts of having a life-threatening illness in a child. And now we, of course, know that PTSD is not just for first responders, it's not just for veterans. Children, too, can experience PTSD, and not just through motor vehicle accidents or violence. It can also happen through having a life-threatening illness. And in some of our early work, we found that full-blown PTSD diagnosis was not super common, but when we broke down the core dimensions of PTSD and looked at hyperarousal symptoms, again, the startle response, re-experiencing or having flashbacks, a lot of these kids were suffering and experiencing these things. So just considering the diagnostic boundaries was not enough to really understand what was happening. And the second thing we did was use neuroimaging to understand how these psychological symptoms can relate to brain development. And we found that kids that reported having more cancer-related post-traumatic stress symptoms had larger volumes of the amygdala, which is an area important for salience processing, for fear responding, highly involved in things like anxiety and PTSD. This was an interesting pattern because we see in animal models that animals that are exposed to chronic stress, we see a similar pattern where their amygdala gets larger and we think more reactive to living in a harsh environment or an unpredictable environment. So I know I've covered a lot of ground. Are there any questions about trauma or some of our work in that area before we move on? Also just wanted an excuse to drink my tea. Okay, so for the second exposure, I want to move on to air pollution, and this is where the CURES Center comes in with our collaborations there. So over the past 5 years or so, my lab has expanded our work in what we consider environmental exposures to not just include kind of interpersonal exposures, but also chemical forms of exposures like air pollution, which we all know, unfortunately, is a growing problem. We've all experienced a couple summers ago the Canadian wildfire smoke. We just had some disastrous wildfires in our, with our friends in Southern California. So this is unfortunately a huge problem. And just like trauma exposure, air pollution is not evenly distributed. It disproportionately affects marginalized communities. What I'm showing you on the left is particulate matter, fine particulate matter air pollution exposure across Metro Detroit, shown in the more warm colors or more air pollution. And PM 2.5 just indicates very small particles, smaller than a single strand of hair. So some that could be easily deeply penetrate the lungs, increase inflammation, and even translocate into the brain directly through the olfactory bulb. So these are highly problematic. And then on the right, what I'm showing you is an overlay of demographics of these zip codes with air pollution exposure being densely located in the same areas where our most vulnerable communities are. And you can see, particularly in Southwest Detroit, where there's a lot of industry, a lot of sources of air pollution exposure. And neighborhoods in Detroit, just like a lot of urban centers across the US, experience really high levels of air pollution. Of course, it fluctuates a lot based on the day or the season. If you were to look at this map on July 4th or July 5th, you might see that it's all red. And Detroit, unfortunately, consistently, again, ranks pretty high in terms of air pollution exposure. Our recent American Lung Association gave us an F grade in terms of our air pollution hazards in Wayne County. So we've known for a long time that air pollution can exacerbate asthma, lead to breathing difficulties, even some cardiometabolic disorders. But in recent years, there's been increased focus on the mental health consequences of air pollution. And this is just one systematic review linking long-term particulate matter exposure on the left to increased risk of depression. And then on the right, shorter term or more acute exposures can even lead to greater risk of death by suicide. So what about kids? Again, we know that children and adolescents are more vulnerable to these effects because of that rapid brain development I showed you earlier, and also because their smaller bodies consume more air per body weight than adults. And also the lung epithelia and other protective layers that could prevent some of these damages from occurring are still developing. So children and adolescents may be particularly sensitive to these effects. So about a decade ago, some collaborators at University of Cincinnati started looking at the psychiatric effects of air pollution in children and adolescents. So I'll show you one study by Kim Yolton and her colleagues who looked at traffic-related air pollution as a proxy of diesel exhaust particles, because we know that living close to I-75, for example, is associated with greater exposure to PM 2.5 through brake dust and exhaust. So using this approach, using residential addresses, Kim found that particularly early life exposure, so think address at the time of birth, was associated with greater depressive symptoms at age 12, which is a really critical transition period into adolescence. She also found similar effects of anxiety scores. And then if you look at current or recent, so at the time of the measurement, there's an increased risk of generalized and social anxiety disorder symptoms specifically. So it's not only recent exposures, they're also seeing effects of cumulative across the childhood period, as well as their recent exposures affecting symptom levels. And taking that a step further, our collaborator Cole Brokamp at Cincinnati Children's Hospital Medical Center did a really interesting study where they examined acute increases in PM 2.5 in emergency department visits for children for any psychiatric indication. He found that an acute increase in PM 2.5 was associated with increased rates of psychiatric ED visits 1 or 2 days later. So it's this really interesting lagged effect. And then if you group by type of disorder, a few interesting patterns emerged. For example, an increase in PM 2.5 was associated with emergency department visits related to mood disorders 2 days later, and for suicidality 1 day later. He also found some interesting moderations by community deprivation, with stronger associations observed between acute PM 2.5 exposure and psychiatric ED visits, suggesting that, again, more marginalized or disadvantaged communities might be greater impacted by these acute fluctuations. So learning all this, my lab was really excited and interested in understanding how PM 2.5 exposure affects the developing brain. What are the mechanisms underlying this association? And just a shout out to Claire Zendel, who I think had to step off if she's not on right now for some advocacy work, which is so important right now. Claire has really spearheaded a lot of this work in my lab, and we could not do it without her. She received an NIH F32 award, so she's a postdoctoral fellow right now. And she came up with this brilliant model for how we think, yay, Graham celebrating, for how we think about air pollution exposure can lead to internalizing disorders like anxiety and depression, as well as other common disorders. And in this model, Claire really focuses on structure and function of the prefrontal cortex, amygdala, and hippocampus as just some of the key players that we know are important for those disorders, and also we know are susceptible or sensitive to air pollution and inflammation. Of course, those are not all the players, but those are some of the key regions we focus on. So in some of her studies, she's using neuroimaging, which I'll show you shortly. And she's also looking at how this may work with inflammatory markers. And we're just beginning to get into more of how this could happen at more of a cellular and molecular level. So in one of her first studies, she used data from the nationwide ABCD study, which, if you don't know, is a longitudinal study of over 10,000 children and adolescents followed annually and biannually starting at age 9 and 10 and across 21 different sites in the United States. And using this huge cohort, she looked at air pollution exposure measured by PM 2.5 from their residential address at baseline and then 2 years later. And she was interested in looking at resting state connectivity of the default mode network, or the DMN, which is a series of brain regions important for internalizing self-oriented thought and frequently implicated in depression. So on the left, she found that air pollution was associated with differential changes in resting state connectivity within the DMN over time. And on the right, she found differences in how the DMN interacts with the dorsal attention network, or the DAN, another area that's involved in orienting attention to important information in your environment. So youth exposed to higher PM, shown in the cell lines, showed lesser increases in within network connectivity, so could indicate less network integration, and then lesser anti-correlation on the right between that network and another large-scale network. So it could suggest maybe a difficulty in disengaging from these two networks. Of course, this is all speculative. It'll be interesting to see if these resting state changes relate to symptom patterns later in life. But then Clara helped us get a CURES pilot project, which we are very excited to share some preliminary results from, to test this in a Detroit sample, or a Metro Detroit sample. So this is a pilot project grant we were awarded originally in 2021, and you can see where we were able to draw children and adolescents as well as a parent from across Metro Detroit. And what Clara did was, again, estimate their PM 2.5 exposure using their residential address, which is a gold standard way to do that in the literature. And what she found, again, was that youth with higher PM 2.5 exposure, I think this was past month, so more recent, was associated with greater symptoms of generalized anxiety as well as social anxiety. So they're actually replicating what Kim Yolton and colleagues have found previously in Cincinnati. She then took that a step further and asked how PM 2.5 relates to brain functioning. And she was really interested in this idea of fear extinction, which is the ability to regulate fear responses in inappropriate environments. And she was interested in fear extinction because we know that that's one of the key processes that we think is involved in risk for anxiety. And it's also relevant for current treatments. A lot of our behavioral treatments, like cognitive behavioral therapy, sometimes include elements of fear extinction. So understanding whether air pollution can affect this association may help us understand the link between air pollution and anxiety, and also develop better treatments or modify our treatments so they work a little better in this population. So what Clara found was that higher PM 2.5 exposure on the left was associated with greater insula response during this fear regulation task. And this is interesting because the insula is a very interesting area. It's involved in a lot of things, but one of the things that it does in the context of fear is it's involved in the expression of fear and also integrating visceral and homeostatic functions. And on the right, she found that the youth with greater insula engagement during this task reported higher anxiety symptoms. So she's finding really unique brain and symptom and air pollution associations. So this was one approach that Claire's been using, again, using these spatiotemporal models to estimate air pollution exposure based on where the child lives, because we think that where they live is a pretty good estimate of their overall exposure to ambient or outdoor air pollution. But we know that kids don't always spend time at home. They spend time going to school or in transit. So she developed a really interesting add-on where she gave kids these personal air monitors, and she had them strap them to their backpacks. Ours have little googly eyes on them. But she was interested in tracking their personal exposures in their day-to-day living. So she had a group of kids wear these around for 7 days. And I'm only showing you a couple of exemplar subjects because we're still working with the data, but she's finding some really interesting patterns. So on the left and the very bottom, you'll see that this particular participant had an increase in PM 2.5 exposure on day 5 that was associated with a concurrent increase in their reported anxiety symptoms. And in contrast, participant 2 had an increase in PM 2.5 on day 4, and they reported anxiety symptom increases 2 days later, so more of a lagged effect. So again, it's really interesting. We're still working on these data. If anyone does have any expertise in more of this within subject distributed lag models, they didn't teach us that in neuroscience graduate school. Please let us know. We'd be really interested in some tips for that or collaboration. She's also mining the data right now. We have brain imaging on day 7. So after this week of exposures, how do those air pollution levels relate to brain functioning and other metrics? So one other thing Claire has done before I move on to the next section is look at inflammatory markers. And things like C-reactive protein and interleukin-6 and others have been linked to air pollution exposure in adults, but not much is known about what's happening in youth. And she looked at lipid and also protein mediators of inflammation. And she found that lipid mediators, particularly the prostaglandins like prostaglandin E2 on the top left, were associated with increased air pollution exposure. And then on the bottom, IL-6, there was an interactive effect where increases in air pollution with higher inflammatory markers were only significant in females. So she's seeing some sex-specific effects there. So again, all this is really preliminary. It's cross-sectional data, but again, does give us an interesting picture of how these brain changes may be explained. So now that we've covered trauma and air pollution, I want to transition to the third and final type of exposure I'll be covering today, and that is cannabis use. So we know that adolescence is a high-risk period for the initiation of substance use, not just cannabis. Experimentation is really common. And cannabis, as we know, is legalized here in Michigan for recreational use for ages 21 and above. Although I guess there's in the public a lot of misconceptions about that it's just legal for everybody, but that's a whole nother conversation. But nonetheless, there's been growing public health concern about how does cannabis use in particular affect the developing brain, particularly for vulnerable populations. Like I was talking with Stephen the other day about pregnancy, but here we're going to focus on adolescence as another period of rapid brain development. So data show that about 25 to 36% of adolescents aged 14 to 17 report using cannabis in the past year. It obviously changes over time and by grade. And I just want to draw your attention to 2020. We were all like holding our breath to see what would happen. Would kids, be home and have less to do, so they'd be using more? Or in this case, maybe they had more parental oversight, so they were using less. But nonetheless, we found there's a little bit of a drop off in cannabis use that seems to be recovering over time. I'm not showing you rates of vaping and other substances here. But what is more concerning, I think, is that there's a decrease in perceptions of risk of using cannabis regularly during adolescence. And actually, the trend is flipping with alcohol for the first time in a very long time. So very interesting. But a lot of research has showed that adolescent onset cannabis use in particular dramatically increases risk for depressive symptoms, cognitive deficits, anxiety, suicidal thoughts and behaviors. And on the bottom left, especially cannabis use disorder, which can affect up to 10 or even 20% of adolescents who use cannabis regularly. So we know that this is a problem and it's very unique to adolescent onset use. So there's good evidence to suggest that cannabis could affect the developing brain. And here's just one study showing that. They looked at gray matter or cortical thickness in different regions and found that even one or two instances of lifetime cannabis use during adolescence is associated with cortical thinning in the prefrontal cortex. We've also shown in some of our pilot data, we had younger kids, so we don't have a ton of cannabis users in our prior studies, but we found that those who use cannabis regularly had greater physiological responses during that fear extinction task. And this is really interesting because there's a really tight association between substance use and anxiety symptoms. And, it's a little bit of a chicken or the egg, like, are they self-medicating, or does cannabis use change the brain in a way that makes these kids more susceptible to later anxiety? That's to be determined. Obviously, I don't have time to go into that whole lecture, but I do just want to do one shameless plug. I'll do another one later, I'm sure. But I had the pleasure of editing a textbook that's coming out this summer that really was an opportunity for me to invite a lot of leading experts and some of my friends to contribute to how does cannabis affect the developing brain. So if anyone is interested, I'm happy to share parts of it or direct you to more information. So there's a lot of interest in whether trauma exposure can increase risk of substance use during adolescence and whether that leads to later substance use disorders, because we know a lot of folks with adult substance use disorders actually began using early life, and also childhood trauma exposure, we know, again, increases this risk. But I don't think this model goes far enough, as we're not considering things like air pollution, which we just thought of, and even diet or lifestyle changes. So I think a broader understanding of the exposome, like what CURES is doing, can help us better understand psychiatric and substance use disorder risk. So last part of my cannabis talk, I want to shift gears a little bit and talk about this idea of secondhand cannabis smoke. And I was at a concert the other week, people are pulling out their vape pens, and I can't help but think, we've come a long way with secondhand tobacco smoke. Like, we've done campaigns. We know that smoking tobacco around your children is not a good thing. But we really have this problem of secondhand cannabis smoke that is not going to go away anytime soon. Michigan loves our cannabis, right? We have a booming cannabis business or economy. So what this study did was they measured PM 2.5 levels in households where active cannabis bong smoking was conducted. And what you'll see right within the first 15 to 20 minutes is that PM 2.0 levels increase by 100 or even 1,000-fold very rapidly. And they decline over time, but obviously it's slower, and it depends on things like whether windows were open or not in the home. But if we think back to tobacco smoke, levels are about 250 microns for a tobacco smoking home. So what we're seeing with cannabis is just knocking that out of the park. And yet there's this misconception that cannabis smoke is somehow safe. So this raises a lot of concerns, obviously, about people that are in the home, pregnant people, children who are inhaling this. And, again, like, we don't think much about, people will be in a car, they'll be smoking, and then they'll pick their kids up from school after. But a lot of that PM 2.5 and other pollutants can linger. And again, we're in Detroit. You can't drive like 20 minutes without seeing a bunch of ridiculous cannabis signs. So again, this problem is not going away. So as Graham alluded to up top, we're really excited that we received funding for a CURES pilot project to work with parents who are using cannabis regularly to see if we can reduce their children's exposure, because we know that cannabis exposure can linger in the homes. And so what we're doing is we are going to be measuring PM 2.5 levels in homes of cannabis using parents and having their child walk around with these air monitors so we can measure their child's exposure as well and see if we can link up their exposures with their parents' cannabis use. And on the left here, this is some of our pilot data. We had one of our unnamed friends wear this monitor while they smoked cannabis flower, shown as flower here, or vaped a cannabis e-cigarettes. And what you can see, again, is that cannabis flower exceeds the daily EPA limit, shown in red, and also vaping. So this notion that vaping is somehow safer is not well supported for PM 2.5, as well as for a lot of the ingredients in e-cigarettes and that liquid, but that's a whole nother lecture again. So yep, we'll be looking at cannabis flower as well as vapor and measuring PM 2.5, and seeing if we can work with parents to figure out how to reduce their exposures. So Claire is one of the MPIs on that project. I'm also really excited that we have Dave Ledgerwood, who's a clinical psychologist and fantastic researcher in my department, who specializes in behavioral intervention development, particularly contingency management. He's done some work with substance use disorders, but also working with parents to help children with obesity. So we're excited to apply his skill set to this unique area. And we're really hoping that it's a pilot, it's proof of concept, but we could expand potentially the same framework to other types of exposures in the environmental health space. So maybe next time I see you all, I'll have some pilot data. Okay, so speaking of positive things, I do want to end on a positive note and talk a little bit about intervention. So what can we do about this? And one exciting system we're targeting is the endocannabinoid system. And because that system was, because it was named because cannabis constituents like cannabinoids, THC, CBD bind to it, it doesn't mean that we don't have this entire system that is involved in stress regulation, appetite, circadian control, feeding behaviors. So this is a really important system for stress regulation and mood as well. And it's increasingly implicated in disorders like anxiety and PTSD. So these are really interesting molecules. They're lipids. We can measure them in plasma, which is the gold standard approach. And what we found is that measuring these levels in kids who reported higher anxiety symptoms had higher anandamide, the moiety on the left here, and lower 2-AG concentrations, another type of endocannabinoid. So this got us thinking of whether endocannabinoids could be potentially a biomarker of psychiatric risk in youth. And interestingly, we're finding that this is tractable to treatments. So we performed a secondary analysis of a randomized controlled trial done by my colleague Jeff Strong at UC. And he had an 8-week clinical trial of escitalopram and SSRI and placebo. And SSRIs are currently the gold standard pharmacotherapy for generalized anxiety disorder in kids. So this is a really unique study. And what he found was that youth who showed greater elevations in 2-AG responded better to SSRI treatment. So they showed greater reductions in anxiety symptoms. So not only are we seeing that kids with higher anxiety have lower 2-AG, but we're also seeing that those who respond to a first-line pharmacotherapy show greater elevation. So we might have something that is kind of tracking the treatment response. We've also found links between anandamide on the left and PTSD symptoms in youth, and PTSD is extremely common. Again, in our Detroit sample, we're seeing about 20% have elevations that suggest a diagnosis, although we're not doing clinical diagnoses here. So it's really interesting because PTSD is in some ways very tractable because of all the psychiatric disorders, we have a clear onset time. The time of trauma is when the pathology begins. If you contrast that with things like anxiety, schizophrenia, ADHD, it's a little unclear when in time those things begin. So that got us thinking, these are cross-sectional data. Can we try to find kids who were just exposed to trauma? And that's not as easy as it sounds, but we borrowed some of the developments from the adult literature where they're looking at people coming into a hospital for a motor vehicle accident and then seeing if they can identify those who will develop PTSD later. And we started thinking about in the pediatric space, what would be very similar to that. And what we thought about was the pediatric ICU, or the intensive care unit. And this is an enormously stressful experience for children and their parents, as you can imagine. And turns out rates of PTSD are also really high following PICU discharge. It can be up to 30% of children, and up to 80% of their caregivers will develop PTSD. And of course, that depends on prior trauma, their baseline mental health. If it was unexpected, does their child show rapid decline? A lot of factors. But we're excited to launch a new study that's looking at endocannabinoids during critical care and seeing if we can predict which children and caregivers will go on to develop PTSD. Do we have a biomarker or a way to identify at-risk children? In another ongoing study, we're looking at a growing mental health crisis in youth, which is suicide. So there's compelling evidence that the endocannabinoid system is also involved in suicide risk. This is measured by some postmortem studies in adults who died by suicide. And in our pilot data, we're finding that youth who have suicidal thoughts and behaviors have higher anandamide than those matched controls without. And of course, this is a very small subset, but we know that suicide is a huge problem. It's the second leading cause of death among youth aged 10 to 14. The most recent CDC data, I think from 2023, indicates that 1 in 5 high school students have seriously contemplated suicide in the past year. And this is a huge problem. We don't have great biomarkers. We don't have great interventions. A lot of kids won't tell anyone about this before making an attempt. So this is work that is led by Samantha Ely, who is now a PhD candidate in my lab. We're very excited for her project, and she's collaborating with Dr. Tamina Shakir, who's a child psychiatrist in our department, who's just an amazing collaborator. So again, next time I see you guys, hopefully I'll have more. So there's a lot of different ways to intervene. Just want to check the time here. Okay, perfect. Graham, I expect you'll just cut me off if I go too long. So from the environmental space, there's a lot of types of intervention. So on the left, we have behavioral, pharmacological, and then also public health interventions. And on the right, I think, again, the environmental health space does a really good job of showing how this can be done. And one example is, I think, of our colleagues at University of Cincinnati who did an anti-school bus idling campaign to reduce children's exposures to PM 2.5 while they're waiting for their school bus. So I think there's a lot of great examples here. As a developmental scientist, I'm always excited about the behavioral, but we've already shown that SSRIs or first-line pharmacotherapies could potentially mitigate some of these environmental risks through things like the endocannabinoid system. And of course, cannabis is one way to target the system, but cannabis we know is harmful for the developing brain, and it also includes hundreds of different chemicals, cannabinoids, terpenoids, and other molecules. So it makes it really hard to target things precisely. So the endocannabinoid system is very complex. Again, I won't have time here to explain all of it, but it's comprised of receptors, the endogenous ligands, and on the bottom, the series of enzymes that regulate their levels, so their synthesis and their degradation. And I just want to point out two Fa and Mag lipase here that are really exciting right now in the clinical trial space for adults, although we just had a couple fail for PTSD, but for other indications, there's a lot of excitement right now about targeting those two molecules, which essentially increase endocannabinoid tone or your endogenous endocannabinoid signaling. And in adults, the number of studies, as well as, again, on clinicaltrials.gov, is just exploding, targeting this cannabinoid system. So we're watching how this is happening to see if we can apply any of this to children. So one pharmacotherapy I also want to mention for kids is cannabidiol or CBD. This is a really common molecule. It's over-the-counter supplement. You've probably seen it. It's non-intoxicating, but it does present really interesting properties. It's anti-seizure, it's antipsychotic, anti-inflammatory, and in some cases anxiolytic as well. It doesn't bind directly to the canonical cannabinoid receptor, but it has really interesting effects on GPR55, the serotonin system, and some other receptors. So during the pandemic, we got really excited about CBD in its FDA-approved form called Epidiolex, which was approved in 2018 for, at the time, 2 severe seizure disorders, because it's an FDA-approved molecule, and it's been shown to be really effective for reducing seizures in kids. So that got us thinking, okay, this is a really interesting way to look at psychiatric effects, because like a lot of kids with epilepsy also can have autism-like behaviors, anxiety symptoms, and other kind of behavioral health problems. So we teamed up with Amy Luat, who is a pediatric neurologist and been using Epidiolex since 2018 to treat her patients with seizure disorders. And we wanted to follow her patients as they newly started on Epidiolex to see if it also affects anxiety symptoms. And what we found was that in this very small pilot study, most participants showed reduced seizure frequency that's consistent with what we know about this medication. And a majority also reported reduced anxiety, particularly within the first week of treatment. So this is really interesting to us. The doses here, I just want to do my little hobby horse that the doses of CBD we're talking about here are nothing like the 10 milligram over-the-counter thing that people talk about that probably have no physiological effect. These kids are taking 15 to 25 milligrams per kilogram per day. So these are huge doses. But we're seeing some signal with anxiety, which is very interesting. Obviously, we have another chicken or the egg problem, like are they less anxious because they're having fewer seizures, vice versa? Is there a direct effect of CBD on anxiety? So we actually are waiting on a pending R61 grant to look at CBD as an adjunct to cognitive behavioral therapy with my colleagues Leslie Lundahl and Christine Rabenak. So again, hopefully next time I see you, we'll have more information. So one last area is behavioral interventions. Again, that's something we always try to look for in kids. And one of the things we're really excited about is exercise. So there's a lot of good evidence that exercise can modulate the endocannabinoid system. And actually, the runner's high, that feeling of euphoria and lower pain, lower stress that some people experience, is not related to endorphins. It actually turns out it's mediated by the endocannabinoid system. There's a lot of really interesting studies there where they block the opioid system and you still get the runner's high effect. But if you do the same with the endocannabinoid system, you don't. So really interesting work. But my former undergraduate student, Shreya Desai, for her very first project, she decided to do a systematic review and meta-analysis, which I do not condone for a first undergraduate project, but she was very stellar. She was really interested in whether endocannabinoids could be modulated by exercise. And turns out the answer is yes. She found that across rodent models, across humans, this includes healthy people, people with fibromyalgia, trained athletes, people with depression, an acute bout of exercise, like 30 minutes cycling, running, yoga, you name it, was associated with an increase in endocannabinoid concentrations, particularly anandamide. Although there is a little bit of variability, but across all these studies, this is a pretty powerful effect. So this is really cool. We have a behavioral way to modify your endocannabinoid system. So we started thinking about whether we can apply this to children, and specifically in the context of fear extinction, which we think is relevant for anxiety risk. So we have a current R01 where we're kind of hacking the system with exercise, but we're having kids run on a treadmill right after they learn that something is safe or undergoing fear extinction. And the idea there is that we think that endocannabinoid boost during that memory consolidation period can increase the ability to recall that that memory is now safe. So we expect them to show lower physiological responses when we bring them back a day later. And we're doing neuroimaging as well as measuring endocannabinoids in blood. And this is a randomized controlled trial. So sounds very technical and kind of weird, but if this works, you could imagine that this could be a really nice way to increase therapy outcomes. So you could have a child go to their cognitive behavioral therapy session and run on a treadmill for 30 minutes right after that, and then maybe they respond better to that treatment, require fewer sessions. So we're really excited by that possibility that this could be something to enhance a first-line treatment that we do know works in some, but not all cases. So I've covered a lot of ground. I hope I have convinced you that adolescence is a really important period of brain flux and also psychiatric risk, and also a time when environmental exposures can have lasting impact. Today, I obviously just gave you really quick snapshots, but I did want to highlight 3 key environmental exposures, and you can think back as to whether you were correct or not about what you voted early on, but all of these we think changes the brain in ways that make some people susceptible to mental but also physical conditions. And then I also mentioned some of our data looking at the cannabinoid system as a biomarker of risk, and also I think a very exciting target for intervention. So one more thing before I wrap up, 2 more things actually. I did want to pause and just say I would be really remiss if I didn't encourage you all, given the current climate, to not only communicate science like we're doing here to our peers, but also to the wider public. I think it's really important, especially now. And there's a lot of ways to do this. Again, shameless plug, we have a podcast where we try to demystify science for the general audience. We try to reduce stigma about brain disorders by talking to people with lived experiences. Love to chat with some of you on this call if you ever want to talk about anything, any of your work. I love that. We love involving students in this, so please reach out. Social media is another kind of necessary evil. Oh wait, sorry. I did want to highlight 2 podcast episodes that I think are really of interest. On the left, we just launched a podcast entitled Pulverize the Stigma of Mental Illness by John Nelson, who has a really powerful story about his life being saved by a clinical trial of deep brain stimulation from Helen Mayberg's group. Really powerful message here. And then on the right, my friend and colleague, Matt Hill, came on and really helped guide a lot of the misinformation and disinformation about cannabis science, which can be really polarizing, but I just find it to be a really science-based conversation, and he's a very clear communicator. So if you want more information about cannabis, highly encourage you to listen to that episode. But as I was saying, social media, necessary evil. I think that's where a lot of these folks are that we're trying to reach. And in my lab, we've even done a TikTok series about the science of CBD led by one of my former medical students, but it doesn't have to be that extensive. Talk about what you do as a scientist, why science is important. You don't have to make an entire series, but I think it's so important in this day and age. And then lastly, be a science advocate. Raise your voice, go to town halls, write an op-ed about how environmental exposures affect your neighborhood. You can do this in 5 minutes through 5calls.org. It takes literally a couple of minutes to talk about the issue that you want, call your lawmakers. They make it really easy. And then another plug is we have Science Policy Network Detroit, or SciPol Detroit, which we created as a platform for students as well as scientists to better communicate to the public and to lawmakers. And this bottom image was from their trip to Lansing last week, where they talked about several science-based issues that they care about. We also go to Washington, D.C. every year. So if anyone wants any resources, we love for you to get involved. But if you want information on how to write op-eds or contact your lawmakers or schedule time with them, please don't hesitate to reach out to us. This is our passion and what we want to do. So with that, thank you so much for listening and for the invite.
Thank you so much, Hilary. That was fantastic. I'm sure there's going to be a lot of people that are very keen to ask questions.
I love it.
Oh, I'm just seeing applause at the moment rather than raised hand. Chris, unmute and go ahead, please.
Great job, Hilary. Really fascinating. My general question is about, you talk about these like 3 different exposures, different areas of research, a common, kind of theme across is anxiety. But so the general question is about sort of like links between these different exposures and ways to analyze these. I guess I'll throw out a couple of ideas and then see if you have others or want to comment on these. But for example, with the trauma exposure and with pollution and PM2.5, I was wondering, like, for example, with the ABCD, like a big study where it's easier, you got a big enough sample to do these more complex moderation analyses. So things like that. Have you looked at any of those kinds of things? And then the other idea is more in the latter 2 things. It really stood out to me when you were talking about pollution and PM2.5 and then cannabis, but then you actually talked about the PM2.5 through the smoke exposure, so like the secondhand exposure. So that got me thinking about, is it really is it more the toxicant from the air, or is it cannabis exposure in another way? So just kind of thinking about the interactions there as well. So yeah, just some ideas that I was wondering about regarding the different exposures and how they might relate and interplay.
Yeah, fantastic question. People are so messy, right? It's so hard to parse these things out. I love what you're talking about though with leveraging big data. I think that will be really useful, like the ABCD study. Clara, I think she's off again, but she's been really interested in exactly what you're describing, like does trauma sensitize you to air pollution or vice versa? Like, how do these things all interact? Because we know, as we were saying earlier, that a lot of these exposures are layered on top of each other, and that those vulnerable communities are being, affected by a number of different things. So how do we parse that out? Yeah, I don't have great answers, but I love what you're thinking about with big data and seeing if we can parse these things and measure them more precisely over time with things like personal air monitors. And then, yeah, the substance use thing is spot on. Like, these kids are, if they're using, they're going to also be exposed to secondhand, smoke. So what do we do about that? How do we parse those things apart? That wasn't an answer. It was more of like questions back to you, but I'd love to continue that conversation. Or if anyone else has thoughts, please. I know we're also running kind of low on time, but I'm going to put my email in the chat if anyone does want to follow up via email. I love this conversation, so don't hesitate.
I have a question, Hilary. Do you think, in terms of intervening with these children, do you think part of the problem is that they know too much? They have access to so much knowledge that there is no, this is going to sound a little woolly, but there's no sense of wonder anymore in childhood. You just think you know everything that you're ever going to know, and that your options moving forward have already been defined.
That's a good question. Kind of hard to answer. Yeah, I guess I'm thinking about it through like the cannabis lens, because a lot of teens, kind of like you're saying, like, they're not dumb. Like, they see that it's legal for adults, their parents are using it, but they're not allowed to use it. And there's this, relatively arbitrary 21-year-old threshold. So I think I think the scare tactics don't work. We all know that. I think hiding some of the facts doesn't work. But I think they are hungry for answers. So if you come with real, like, here's what we know, here are the risks, here are the potential, benefits, just educate yourself on why you're using, like, what are the risks, can you delay, can you use in safer ways? And I do have some nice resources that I'm happy to share with this group about how to talk to kids about cannabis, which is not easy, but yeah, I don't know, for other topics, I'm not really sure though. What do you think?
Well, I'm not sure. I mean, I used to give talks over in Windsor, and I was just as terrified as I'm sure people are here about how much 9-year-olds know about drugs of abuse, and B, how the pattern of usage has so violently changed. I just don't think I think we're still failing them in terms of talking to them soon enough about these issues. Another question I had, actually, Hilary, was you did actually mention the gut microbiome. I mean, you talked about the fact, we have knowledge that we've always had about substances going in the nose and maybe going to the olfactory bulb. You also potentially through the eye, through the optic nerve. But now we've got a whole panoply of what we thought were neurological conditions that appear to be modified by what's going on in the gut microbiome. And I wondered if that's something you're thinking of pursuing.
Yeah, we've thought about it a little bit. I do not have expertise in that area. So if someone wants to help us with that, I think you're spot on. I think that's another uncharted territory.
Mike, go ahead.
Hey, Hilary, just a quick question. So a lot of the clear, like the Superfund folks here, are really interested in utero exposures or even preconception exposures. Yeah. And I thought it was really interesting with that one Cincy study where they saw the significant effects in the early stages, the earliest exposures. Is anyone trying to go back in time and look at where should we intervene? At the maternal side, preconception, or early stage? Has anyone looked at moms and dads that have moved and then looked at the offspring? I'm just curious.
Yeah, great questions. I don't know everything the Cincinnati group is up to, but I'm sure they have a ton of longitudinal studies. So I think you're probably circling around what they do. Also for cannabis use, again, I didn't have to talk about, the pregnancy data, but these cannabinoid receptors are established at week 7 or so of gestation. And a lot of moms find out that they're pregnant around that time or even after. So think about a lot of these moms using cannabis and how that's affecting the developing brain. So we're starting to look into that a little bit. We've used, as Chris mentioned, the ABCD data to look at prenatal cannabis exposure. And again, we have the sample size to be able to parse that out from tobacco or nicotine exposure versus alcohol. So we're finding some really interesting long-term patterns on brain development measured at age 9, 10, and 11. But yeah, I think that's really the next question is like, how early does all this happen? When do we need to be intervening and how?
Thank you.
Thinking also about earlier intervention, Candy in the chat from the Mott Center is asking though about sex dimorphic patterning in your study, and are there differential effects of cannabis on the boys and the girls' brains that you've seen?
Yeah, so we've only started to look at adolescent cannabis use and how it relates to our outcomes. I think some of the literature has found sex differences and certainly age of onset of use. The earlier and the more frequently you use definitely are associated with worse outcomes, with greater cognitive dysfunction, greater risk of anxiety, suicide, things like that. So that's something we're definitely interested in exploring.
Well, I remember from my stem cell experience, there were sex differences in neural stem cell populations in the developing brains, and that would be in the hippocampus. So I don't know if that's on your map to be looking for differences there, but might be very interesting to work out whether that has a role.
For sure. Yeah, that's fantastic.
Okay, well, we're at 1:31, and I like to keep things on time. It's been a fantastic presentation. Everybody's really enjoyed it. So, ladies and gentlemen, let's thank Hilary once more for a fantastic presentation.
Thanks, everybody. It was a pleasure. Bye.
Thanks, Dr. Marusak. And just so everybody is aware of our upcoming seminars, I'm sharing my screen now. I hope you can see the schedule. Next week, we have Robert Goodwin and Aaron Bunting. They work with us here at CURES, and their presentation is about a geographic information system that we make use of here called MIDAS. Will be a fascinating explanation of how we use that in our environmental health research here at CURES. So please be sure to join us next week, Thursday, 12:30. Hopefully we'll see you then. Thank you very much.
Thank
Thank you.